We are going to continue the topic reproduction in organisms. We have already talked about it in our previous class. So this is our second class, and we'll be starting binary fission today, right? So we were talking about the different types of asexual reproduction, and the first one is binary fission. So what is binary fission? It is a process in which the parent organism divides into two equal halves and each half behaves as an individual organism, right? Here the parent as a whole forms the reproductive unit, and it continues to live along its offsprings. Because of this, the parent is said to be immortal, right? Organisms that undergo binary fission are said to be immortal. What is immortal? Something that never dies is called as immortal. Now, these organisms are obviously living, and so they will obviously die. Kintu, no doubt they die. Kintu yate ami ki likhisu that the parent will divide into two halves. Aru ami ki nedekho parent or dead remains kini dekha na pao, and that is the reason why we are using the word immortal, right? The parent obviously dies, but we don't know, we do not see its dead remains, and so we are using the word immortal. Next, enupa process is seen in binary fission. Ami kod dekha pao. It is seen in Monera, Protista, etc. In case of Monera, what is the best example that we are talking about is bacteria. Protista, we are talking about Paramecia, etc. So binary fission is a type of asexual reproduction. It is seen in lower organisms, seen in the kingdom of Monera and in the kingdom of Protista. Now, what is Monera? What is Protista? I think we remember we have talked about Whittaker's 5 kingdom classification when we were studying about the chapter in class 11, right? So, whenever we talk about Monera, we know that Monera is said to be the kingdom of prokaryotes. Our target amar ki asile bacteria asile, eubacteria asile, cyanobacteria asile. So these were included under Monera. Similarly, the next kingdom was Protista. When we talk about Protista, the first line that we remember is it is the kingdom of unicellular eukaryotes, right? Protista, we have talked about some examples. For example, kiki asile, we have talked about diatoms, we have talked about dinoflagellates, euglena, etc. So Protista, another point that you have to know is it is also called as a junk box. Why is it considered to be as a junk box? Because this is the only kingdom where you see all sorts of organisms. All sorts of organisms means what? We see certain plant-like organisms, we see certain animal-like organisms, and we see certain fungi-like organisms. All these different type of organisms belong to the same kingdom. So it is considered to be a junk box according to Whittaker's 5-kingdom classification. So we are not going to cover the details of Monera and Protista. This is something that we have done in biological classification of class 11. Coming back to our topic, so we are talking about binary fission. It is a very simple process. In this case, what happens is we have the parent. This parent will divide into two, and ultimately it will form two daughter individuals, right? Now, depending on something that is called as plan of division, it's a binary fission or ki hoy that there are different types of binary fission that is seen in different types of organisms. So we will be discussing about the types of binary fission. Types of binary fission. Binary fission, depending upon the plan of division. And when we are talking about division, what we are talking about is obviously karyokinesis and cytokinesis. These are the two terms. What we understand from these two terms is when we talk about karyokinesis, it is nothing but the division of the nucleus, and when we talk about cytokinesis, it is the division of the cytoplasm. Whenever a cell divides, these two processes is something that always occurs, and which step occurs at the beginning? It is always karyokinesis that occurs, and it is later followed by cytokinesis. So this parent cell, if it undergoes binary fission, depending upon the plan of division, binary fission has been categorized into three types. So there are certain organisms that would undergo simple binary fission, that is also called as irregular binary fission. Some organisms will undergo longitudinal binary fission, and while others would undergo transverse binary fission. So depending upon the plan of division, three types of binary fission: simple, longitudinal and transverse. We will be talking about the first one, that is simple binary fission. So simple binary fission is also called as irregular binary fission. Why are we using the word irregular? The answer is, in this type of organism, the division or the plan of division is not definite. Simple binary fission. So what we'll write? The organism that undergoes simple binary fission can divide in any plan. They do not have a specific plan. Je lengthwise is a divide koribo lagibo ba transversely is a divide koribo lagibo tedikwa eku kotha nai. Juntu side mon jai tahate divide koribo paribo. So it is seen in, and the best example, the most common example that we talk about is in case of amoeba. So what happens in amoeba? If the amoeba have to go for binary fission, what it does, we are going to depict it with the help of a diagram. Suppose this is amoeba. Right? What is this? Or what is this? Nothing but pseudopodia. Right? What is this? It is the nucleus. So during the time of division, this parent amoeba, the first step that happens to it is it will minimize its pseudopodia. Right? Next, once the pseudopodia has been minimized, the nucleus starts elongating. We know karyokinesis is something that always occurs at the beginning, and so this nucleus will start elongating and it will proceed towards karyokinesis. And ultimately, the next step is the cytoplasm will get constricted and ultimately 2 daughter offsprings would be produced, or 2 new amoeba would be produced. Right? So this is what happens during simple binary fission that is seen in case of amoeba. We'll start again. So what happens is, we are talking about the parent amoeba. They have the pseudopodia. During the time of reproduction, they will minimize their pseudopodia, and after that karyokinesis will start, and this will lead to the elongation of the nucleus. The nucleus will divide into 2 nuclei, and ultimately at the same time the constriction of the cytoplasm will also occur. Once the constriction occurs, or if the constriction deepens, then it will split into 2 organisms. One thing that we have to remember here is, when we are talking about the constriction of the cytoplasm always constricts at right angle to the elongated nucleus. If suppose the nucleus is elongating in this way, the constriction of the cytoplasm always occurs in this way. If suppose the elongation of the nucleus occurs in this way, then the constriction will occur in this way. So constriction of the cytoplasm is always at right angle to the elongated nucleus. So this is what is called as simple binary fission. Why is it simple? Because the plan of division is not definite. I think we understood. Next, we'll go to the second one. What was the second type of binary fission? It is called as longitudinal binary fission. So here the plan of division will be lengthwise. So what we understood is cytoplasm will divide longitudinally. So there are certain organisms which will show longitudinal division. This is a process that is seen in case of flagellates. We have a term called as flagellates. We have another term that is called as ciliates. What is the difference between flagellates and ciliates? If the organism have flagella, it is called as flagellate. If the organism is surrounded or have or moves with the help of cilia, it is called as ciliates. It is seen in case of flagellates, and one example of a flagellate could be euglena. Now euglena is something that we have already studied. What we know, it is a link between plants and animals. Euglena belong to which kingdom? It belongs to does it belong to Monera or it belongs to Protista? Obviously, Euglena belongs to Protista. It is a link between plants and animals. Why is it called as a link between plants and animals? Because it shows certain characteristics of plants and it shows certain characteristics of animals. Now, what happens in Euglena? We'll just see with the help of a diagram. So, what is the second step? Again, karyokinesis is something that will occur constriction of the nucleus and finally the constriction of the cytoplasm will also deepen and ultimately two new euglena would be formed. So this is what is called as longitudinal binary fission. In this case, what we have seen, we have the parent cell. In this case, the cytoplasm will always divide lengthwise and so it is called as longitudinal. It is first, what happens is karyokinesis followed by cytokinesis, and once the cytoplasm deepens, ultimately it will form two daughter euglena. So this is what is called as longitudinal binary fission. So if the cytoplasm divides lengthwise, this is called as longitudinal binary fission. The third one is transverse binary fission. There are certain organisms in which the cytoplasm splits or divides transversely. It is seen in case of ciliates. Example, paramecium. So, what happens in paramecium? So, if you are talking about a paramecium, it is surrounded by some fine hair-like structures and these are called as cilia. And we know they have a pair of nuclei. What is this? Contractile vacuole. This one, macronucleus. This one is called as micronucleus, and this is the posterior contractile vacuole, right? So this is the first step, or this is considered to be the parent paramecium. What will happen to this? The nucleus will elongate. Elongated nucleus. This was the macronucleus. This macronucleus will undergo mitosis, and micronucleus will undergo mitosis. Ultimately, this constriction will deepen I'm just drawing a very rough diagram. The proper diagram has already been given in your notes. Ultimately, we will see the formation of two daughter paramecium. So in this case, what we have seen is transverse binary fission is seen in case of ciliates, and in case of ciliates, the best example that we are discussing is paramecium. This paramecium will divide transversely, and so the name has been given as transverse binary fission. So this was all about the first type of asexual reproduction, that is called as binary fission. Now, fission is of two types. We have talked about it in the last class. What are the two types of fission? It is binary fission and multiple fission. The name binary has been given because the parent organism divides and forms two organisms. The next type of fission is multiple fission. Why is it called as multiple fission? Because here, as the name suggests, we will be having, or we will obtain, multiple number of offsprings from the same parent, right? So multiple fission, as the name suggests, process through which it's a very simple sentence that we are writing, process through which many offsprings are formed or produced from a single parent simultaneously. It is seen in, or this type of a process is seen in, examples could be amoeba, plasmodium, etc. Remember, we have discussed about amoeba before. amoeba, we have taken the examples in simple binary fission. What we need to remember is when we are talking about lower organisms, these type of lower organisms does not only have one type of reproduction, but they might reproduce by different methods. It is only the higher organisms that they stick to only a particular type of reproduction. Amoeba obviously is an example of a lower organism, so it can have different types of reproduction. So in case of amoeba, under natural condition or favorable condition, it will undergo binary fission, and under unfavorable condition, it always undergoes multiple fission. So this is something unique to amoeba. So normal condition, or when all the conditions are favorable, they will reproduce with the help of binary fission. But when condition is unfavorable, then they will go for they will undergo multiple fission. So multiple fission, so in case of binary fission, one amoeba will produce two amoeba, but in case of multiple fission, from one amoeba we can have many number of offsprings at the same time. So simple binary fission we have already discussed. What happens in multiple fission? So we will see during unfavorable condition, the amoeba withdraws its pseudopodia and it secretes a three-layered cyst wall around itself, and this process is called as encystment, or it is called as encystment condition. So we are talking about this amoeba. Right? So what it will do? It will withdraw its pseudopodia, right? Or we can write pseudopodia withdrawn. Next is it will form a thick layer amongst itself or around itself, and it's almost like a cyst, right? What happens next is, during this unfavorable condition, within this cyst, what is this nucleus? This nucleus will divide and redivide and form many nuclei while staying within the cyst itself. Under favorable condition, the cyst wall will break and this spores will liberate or get released, and these pseudospores would behave as independent amoeba. Right? So this is what happens during unfavorable conditions. Cyst wall breaks. And when does the cyst wall break? During favorable condition. So amoeba during unfavorable condition, what does it do? It first minimizes its pseudopodia. It forms almost a spherical structure. It secretes a thick wall around itself so that it can remain protected during the unfavorable condition. And while they are within this cyst, the nucleus will divide and re-divide, and ultimately within the cyst wall many small amoeba would be formed, and this amoeba at that time would be called as what? Pseudopodiospores. Once the favorable condition arises, then the cyst wall will break and this will get released, and this pseudopodiospores that got released will ultimately behave as independent amoeba. So what we have seen is, from one single amoeba, what we have seen is the production or formation of many offsprings at the same time, and so this is what is called as multiple fission. As we have discussed, multiple fission is not only seen in case of amoeba, but it is also seen in Plasmodium. So we will be talking about Plasmodium now. I think we understand what a Plasmodium is. We have heard about a term called as, or we have heard the name called as Plasmodium vivax. What we remember, it is something that causes what? Malaria, right? So in case of Plasmodium also, multiple fission is observed. So the next example, multiple fission in Plasmodium. Informations about Plasmodium, what we know? Causes malaria, right? What is it? It is nothing but a obligate parasite. So when we talk about parasite, duta word we know, one is called as obligate and the other is called as facultative. Obligate, I think you remember some words called as obligate aerobes, facultative aerobes, obligate and facultative. Obligate mane ki strict, means what? If the host is absent, then that organism will not survive. That is what is called as obligate parasite. So what happens in Plasmodium? In Plasmodium, multiple fission occurs in two steps or in two stages. It occurs in something called as sejoint and oocyst. What is a sejoint? It is a unicellular structure found in liver and RBC or red blood cells of humans. And multiple fission occurs in two cells. One is called as sejoint and the other is called as oocyst. And what is oocyst? It is nothing but encysted zygote. And where will you find this oocyst? In the stomach of the female Anopheles mosquito. Right? So in case of Plasmodium, Plasmodium undergoes multiple fission. Multiple fission কোট হয় দুটা সেলত হয়, এটা হল sejoint আর এটা হল oocyst. This cell is found within the human beings and this cell is found within the stomach of the Anopheles mosquito. Now when multiple fission occurs in the sejoint, the process is called as sesogamy, and the offsprings that are produced are called as merozoites. And when multiple fission occurs in the oocyst, the process is called as sporogamy, and the offsprings are called as sporozoites, right? So up till here what we have learned is fission could be of two types, binary fission and multiple fission. Binary fission, two offsprings are produced at one go. Multiple fission, many offsprings are produced simultaneously. Multiple fission is seen in, we have discussed about two examples, that is in case of amoeba and in case of plasmodium. When does amoeba undergoes multiple fission? It always undergoes multiple fission during unfavorable condition, but during favorable condition it always reproduces with the help of binary fission. Similarly, in case of plasmodium, we have talked about plasmodium. Multiple fission occurs in plasmodium in two stages, that is the sejoint stage and the oocyst. Where will you find the sejoint? Inside the human beings. Oocyst is nothing but the zygote and it is found inside the mosquito, right? And the process of multiple fission in the sejoint is called as sejogamy, and the offspring that is called as merozoites, and in this case the process is called as sporogamy and the offspring that are produced is called as sporocytes. So this brings to an end about fission. So we were talking about the types of asexual reproduction. There are almost 5 to 6 types of asexual reproduction. The first one was fission. Fission divided into simple, or sorry, it was divided into binary and multiple. So this is all about fission. So we'll be going to the next one. What is the next one? Second to ki asile. The second type of asexual reproduction was budding. So in this type of asexual reproduction ki hoy, a bud is produced in the parent and this bud behaves as a offspring. So a and this bud will behave as an offspring, right? This type of asexual reproduction is seen in yeast and it is also seen in animals, right? Yeast are animals. In case of animals, we see two types of bud formation, that is exogenous and endogenous bud formation, right? Exogenous bud formation is seen in case of hydra and endogenous bud is seen in case of sponges, right? So at first we'll be discussing about yeast. What is yeast? It is an example of unicellular fungi. Generally, fungi are multicellular, right? But one example of a unicellular fungi is yeast. So what happens in yeast? If suppose we are talking about a yeast like this, what happens is formation of bud or a protruded structure, and that structure is called as bud. And finally, the bud will be more distinct the nuclei will divide, one nuclei will go to the bud, and ultimately we will see a structure like this, right? So yeast is an example of budding. In this case, what happens is the parent organism will develop a protruded structure or little slight enlarged structure. This will ultimately form a small compact part, and that is what is called as the bud. This bud, ultimately the nucleus will divide, and it will divide into two nuclei. The bud would be formed, and this bud will behave as right. So sometimes bud could be one, or sometimes they can form chains of buds also, and later they will get detached from the parent body, and then they will germinate into an individual organism. So this is an example of budding in case of yeast. So if an organism or reproduces with the help of a bud, it is called as budding. So what is a bud? Nothing but a protruded or a small compact structure that develops on the parent body itself. Either the bud is formed on the surface of the parent, or the bud is formed inside the body of the parent, and accordingly will have different terms for that bud that is produced. In case of yeast, this is the process. So the next is, how does budding occurs in case of animals? So budding is not only confined with fungi or in case of yeast, but we have examples of animals also. In case of animals, budding could be of two types. One is called as exogenous budding, and the other is called as endogenous budding. And what is the meaning of both the terms? What we understand, if the bud is formed inside the parent's body, then it is called as endogenous. If the bud is formed on the surface of the parent body, it is called as exogenous. So organism tor bhitorot jeri bud ba tenuka horu horu structures develop kore, tak ami ki boli ko endo. And what we understand by endo, it is nothing but internal, and what is exo? It is external, right? So an exogenous budding or exogenous buds, they will be producing external buds, and endogenous budding is a process through which they will be producing internal buds. Right? So in case of animals also, we see two types of budding. One is endogenous and the other is exogenous. In case of exogenous budding, the best example that we discuss is of hydra. It is seen in hydra. So what happens in hydra? We'll just depict it with the help of a diagram. So suppose this is a hydra, they will have the tentacles and this is the mouth. So at the time of reproduction, what would the hydra do? They will form a small structure that is called as bud. Later, the bud will become more distinct and ultimately it will get detached from the parent body. This was the bud, or it got detached from the parent body, and this bud will behave as a new organism. So external budding or exogenous budding is seen in case of hydra, where the bud is grown on the surface of the parent. This bud will gradually keep on growing, it will finally take its own structure and then it will get detached. Once it gets detached, it can form a new organism. This is called as exogenous budding. The next example is endogenous budding, and we know, we have already talked about its example. Endogenous budding hami ko dekha pao. The best example was in case of sponges, right? Endogenous budding, right? So yate ki hobo buds bilak parent or parent organism tor bhitorot form koribo. It is properly seen in sponges. In case of sponges, the buds that are formed are called as gammules. I think you remember we have talked about it in our first lecture, the difference between gammules and gamma cup. So in case of sponge, how does the gammule look like? So we have something like this. So we'll just give a rough diagram of a gammule. So they will have on the surface, they will have some spine or spike-like structures, and this would be called as spicules. So this would be the mouth. These small cells would be called as archaeocytes. This is the outer membrane. This is the inner membrane, and these are called as spicules. These are nothing but spike-like. So this is the structure of a internal bud, or it is called as gammule in case of sponges. So in case of sponges, we have seen the structure look like this, and inside this cavity we will have small cells, and these cells will be called as archaeocytes. And once this mouth, or it will be better if we use the word micropyle, right? So once this get matured, what happens is the micropyle will get open and the archaeocytes will come out, and these archaeocytes will behave as or will develop into fresh colonies of sponges. So in this way, from one organism they will form many sponges. So this was the formation of endogenous bud in case of sponges. So up till here what we have discussed is, we have talked about the second type of asexual reproduction, that is budding. The third type of asexual reproduction would be fragmentation. Next would be regeneration. We have to talk about vegetative reproduction also. So I think for today we will be completing up till here. In the next class we will be talking about the third type of asexual reproduction. Thank you.